So firstly, I'm sorry I couldn't be there in person, but I certainly appreciate this opportunity to participate, at least remotely. As you can see, we have sort of the same name for our project, and doesn't have the 3D on it. But we've been working on this for several years, and I'm really excited about the prospect of joining a larger community to push the technology forward and to really do great things for computational cell biology. The project has been funded since 1998 by NIH. As you can see, we have a good-sized user base. The first number is probably a little bit exaggerated in the sense that most of the registered users probably haven't really done anything serious with virtual cell. They just register and try to see what it's all about. But we do have almost 4,000 users who've actually gone in and used virtual cell to build models and run simulations. We are built on top of a database, so the models can be stored here at the University of Connecticut Health Center. And we have a large number of models and an even larger number of simulations, because you can have many simulations per model. If a model leads to a publication, we encourage our users to make the model completely publicly available. And so we currently have 563 publicly available models. The people behind Virtual Cell, I really like to acknowledge right at the beginning of every talk. These are the three scientists who are my collaborators in the project. They're really the brains behind it. And Jim Schaff has been in on it since the beginning. Boris and Jan have joined him shortly thereafter. They've been really driving this project from the start and have created what it is today. So I'd like to tell you a little bit about what Virtual Cell is, what it can do, and then what the challenges for the future are. So that equation on this slide is sort of a summary equation for the kinds of problems that Virtual Cell can currently deal with. The change in the concentration of a species I as a function of time is given by the divergence of a flux, and the flux can include advection, it can include a velocity or a flow, as well as diffusion. And then, in addition to that, we have a source term, R sub I, corresponding to either the creation or the destruction of our molecule I as a function of other molecules or other states of the system. So it can deal with reaction, diffusion, advection. It can also deal with electrophysiology. We can have membrane potential and currents in the model. It can deal with all the way up to 3D geometries, including geometries derived directly from microscope images, like the one that's playing up in the upper corner. I hope you guys can all see that. And that little simulation was of a membrane FRAP experiment. It's geometry derived directly from a stack of confocal images. The software can create simulations either using deterministic solvers in as either ODEs or PDEs, as well as stochastic simulators either for spatial or non-spatial models. It can do parameter scans, and at least for ODEs, it can do parameter estimation. And as I mentioned before, it's built on a database of models.
Now, it doesn't attempt, despite the similar title of the project, it doesn't attempt to do, at this stage certainly, whole cell modeling. We really model specific mechanisms or specific processes that might be occurring inside cells. And the class of biological problems are listed: signaling and metabolic pathways, intracellular trafficking, ion channels. We also are very often used by microscopists to try to analyze their experiments that use fluorescent indicators or probe distribution, FRAP experiments, FLIP experiments, FRED experiments, etc. This is a screenshot of the software user interface, at least one part of the software user interface. And the main reason that I'm showing you this is just to give you some feeling for the structure of the software. This biomodel navigator pane that I'm showing you here is shows that at the top level of what we call our biomodel, we have what we term the physiology. The physiology contains all the molecules, all the topology of the system, the membranes and what the membranes enclose, the different compartments. It contains the reactions, how the molecules interact or how they go through membranes. So we have the reaction diagram highlighted here, and so you see the reaction diagram shown on the right for this particular model. Importantly, the physiology is the top of the tree, and underneath that we have a series of applications. This particular model has 10 applications. Each application can invoke a different kind of solver, for example, stochastic or deterministic. It can also use different geometries or different geometry specifications, different initial conditions, sort of virtual experiments. So this is a very powerful aspect of the software because it does allow you to exercise a particular physiological model in many different ways in terms of different mathematics. So the mathematics really correspond to these applications, each of which is combined with the physiology. And then the other thing I wanted to highlight was the database pane, which is where you call up saved models. So this is what you would see if you were searching for a model in the virtual cell database, but it also allows you to access public models in biomodels.net, as well as pathways through Pathway Commons. This allows you to get to various pathway databases as a starting point for building well-annotated models. So as I mentioned, we can do 4 different kinds of models. We can do simple ODE models. This is a physiology representing nucleocytoplasmic transport, where a molecule is moving out of the nucleus and coming into the cytosol. So here's the molecule coming out of the nucleus, and here it's appearing in the cytosol. And so we can solve this using a compartmental approach where we know what the membrane surface areas are and we know what the volumes are. So we do have geometry information in there, but it's all ODEs. We can also solve it stochastically. We can then apply this geometry or apply this physiology rather to a real geometry. And so this is showing you that we can solve the PDE. So this is a PDE solution. Usually the PDE solution is a little bit different than the ODE solution at a particular point, although it might converge if all of the processes are fast enough. And finally, we can also do spatial stochastic simulations. So here's a particle-based simulation in the same geometry as for the PDEs. So all of those different kinds of models can be run off of the same physiology and are actually all part of the same overall biomodel structure. I wanted to give you a more detailed example of the kinds of simulations we can run. This should be a familiar diagram to cell biologists. It's a diagram of dendritic actin nucleation, a cartoon diagram that has been published in many of Tom Pollard's papers. And we've translated that into a virtual cell model. This is the corresponding virtual cell network diagram. And a lot of it is reduced, but it retains a lot of detail. And the real advantage of this system is that a lot of the numbers have been measured, many of them, of course, by Tom Pollard. So these are the mechanisms that are encapsulated in the different parts of this reaction diagram. So we've really put together a fairly detailed model of actin dynamics. We can then solve that model using ordinary differential equations for an average size cell, but we can also project it onto a geometry. And this is not a microscope-derived geometry. This is an analytical geometry that we created to sort of represent a cell migrating along a surface. And we've activated actin polymerization at the membrane, which is what happens in real life, along this sort of graded region of the activator molecule, the nucleation promoting factor called NWASP. So this is just a color scale showing you the graded distribution of NWASP that is used as sort of a starting point for actin. These movies that are being played on the left here show a cross-section through this 3D geometry where we're looking at just a selection of several variables that come out of the model. The model does include advection or velocity, and so the first pane here shows you the velocity distribution. The movie here in the second pane is the distribution of monomer, the most active monomer, average filament length, and filament concentration. So again, just to give you an idea of the kinds of things we can do. This shows the steady state values of several of those same parameters, including the total F-actin at steady state after the activation of the tip. And this is showing the bottom XY plane and the middle YZ plane. Sorry? Okay. So this is the most important part. You know, what about whole cell models? How much further do we have to go? The actin model that I showed you actually took 10 days to simulate. It had 60 variables. It was in that fairly complex 3D geometry. So there's a lot that we need to do to improve that. And obviously, if we are ever to model everything in a cell, we've got a long way to go. I guess one of the things that I want to emphasize is that in addition to biochemistry and signaling, we also have to worry about things like changing morphology. We have to worry about mechanics. We have to worry about intracellular trafficking. These are things that aren't very often considered in systems biology, but clearly are central to the actual function of real cells. Additional challenges are, obviously, we've been talking about all day how we need lots of experimental data to constrain models, and also experimental data that would be driven by the models and that would be used to validate models. Tremendous diversity of cell types adds to the challenge of whole cell modeling. And then obviously we have the multi-scale problem Cells by themselves are not enough. We need to understand how they interact to make the organism work, and we need analysis tools. If we ever do succeed in getting a fully functional whole cell model, how are we ever going to actually get any useful information out of something so complex? So the analysis tools are critical and need to be developed at the same time. And finally, we have to keep in mind that we need software and data that is going to be accessible to the modeling and biomedical research community. Thank you.